The final points in your pharmacodynamic section in the syllabus review rapidly signaling mechanisms. These are things that are required to understand the molecular effects of drugs. However, they're most often reviewed in detail in your physio and biochem. Once again, the basic sciences to the clinical world, of course. Now, when we look at signaling mechanisms, as a quick review, you know that receptors can be intracellular or within membranes. Intracellular receptors, classically, are going to be receptors to either intracellular compound or certainly from the outside to lipid-soluble compounds. Classic example of an intracellular receptor, a steroid receptor. Steroids, as lipid-soluble hormone, have easy access into the cell to bind to those intracellular receptors. And intracellular receptors generally belong to a group of molecules that we refer to as transcription factors. When the hormone binds to the intracellular receptor, the complex of the hormone and receptor then translocate to the nucleus of the cell, and the receptor portion of the complex will bind to specific sequences in our DNA. Those sequences are known as enhancers, and the binding to enhancers by those specific transcription factors will result in the responses associated with these hormones. Now, because we're talking about a change in gene expression, that effect is not something that will occur extremely fast in the human body. It might require hours, sometimes days, before we will actually see those responses full-blown. And steroid type of responses follow that very much. You review a lot of these steroid responses again in physio. Otherwise, if you want more information about transcription factors and where they bind in the DNA, I would refer you to your molecular biology section of the biochemistry syllabus, particularly the chapter about genetic regulation. And you will encounter a table very much describing those receptors. One key point you'll see are that steroid receptors belong to what we call zinc finger proteins, essentially having a motif of finger-like projection of amino acid sequences that are stabilized by zinc, allowing those fingers to fit within the grooves of the double DNA helix. Well, that's the deal with intracellular receptors. Does it apply to pharm? Of course it does, since we use steroids as a classic group of drugs, either for anti-inflammatory responses or for immune suppression. Of course, besides that common use of steroids, there's always the potential of using steroids in people who have too little steroids, therefore as replacement therapy. More often than not, though, we'll see that the receptors we love to interfere with in that syllabus will be membrane receptors. And there are two broadly different types of receptors that drugs will love to interfere with. Receptors that may be directly coupled to ion channels, and receptors that are linked to G proteins to cause the actual responses. Receptors coupled to ion channels will tend to give just about immediate responses. The idea being the drug, by binding to a receptor, will either open or close channels that would allow positive or negative charges to flux between inside and outside the cell. Those changes in positive or negative charges immediately change the membrane potential of the cell, resulting in potential response on the spot as the drug is given. Very quick responses. Some examples of membrane receptors directly coupled to ion channels could be a nicotinic acetylcholine receptor, where opening an ion channel to sodium allows cells to be rapidly depolarized, and the result in their firing. For instance, a muscle cell may contract, a nerve cell may be depolarized and release neurotransmitters this way. Receptors linked via coupling proteins, particularly G proteins, are going to be yet one of the even more common mechanisms by which drugs will cause their effect at the molecular level. And you see a diagram showing you cyclic AMP, IP3, diacylglycerol, and calcium as second messengers. This diagram is actually pulled out of your biochemistry syllabus and reminds you the G protein coupled receptors and their pathways. This is a classic diagram that has always been high yield on your exam, so follow it well and see it with me. First, to the top left, you see a classic G protein linked receptor. G protein linked receptors have a structure that is not to be missed on the exam. Those are proteins that span the membrane 7 times. Each of the little dynamite stick on the actual diagram representing one of these spanning membrane sections. Those receptors that have 7 transmembrane domains in turn are coupled inside the membrane to G proteins. G proteins are trimeric. They have 3 subunits called alpha, beta, and gamma. And they're called G protein because the alpha subunit binds to GTP in order to do its job. GTP is what gives energy to the G protein to then communicate with an enzyme attached to the intracellular side of the cell. In the cyclic AMP system, the G proteins interfere with adenyl cyclase. And adenyl cyclase is an enzyme that takes ATP and makes cyclic AMP. So what happens with drugs interfering with that system? Imagine the arrow being the drug at the top of your diagram. The drug binds to the outside face of the cell, binds to the receptor, changing its shape. The change in shape in turn tickles the G protein and turns it on. That G protein in turn talks to adenyl cyclase to change the levels of cyclic AMP inside the cell. If the drug is construed as the first messenger, then cyclic AMP levels will become the second messenger in the response system, in the signal transduction system in that cell. Now, G protein wise, there are 2 types of G proteins in the cyclic AMP system, GS or GI. Whereas a GS stimulates adenyl cyclase and increases cyclic AMP, a GI inhibits adenyl cyclase and lowers cyclic AMP. Either way, the change in cyclic AMP level will result in a cascade of enzymatic events that will result in the response at the cellular level. Cyclic AMP interacts with protein kinase A. Think of A for AMP or A for adenyl cyclase. All protein kinases phosphorylate substrate, and the phosphorylation of other proteins result in a change in their activity. For instance, on your diagram to the bottom right, protein kinase A might have dephosphorylated proteins, and that might have resulted in turning them on or turning them off. These proteins might be enzymes, might be ion channels, or on the left side of the diagram might be the Krebs protein. A Krebs protein, when phosphorylated by protein kinase A, then translocate to the nucleus and binds a DNA region called a CRE, a Krebs response element. Krebs protein is therefore an example of transcription factors turned on by phosphorylation, and by binding to CREs, Krebs proteins can alter gene expression. Thereby it shows you that in a cyclic AMP system, drugs that would work this way have 2 types of effects. Some effects might be immediate and stem from phosphorylation of enzymes changing metabolic function of cells, phosphorylation of ion channels changing conductance through these ion channels, but also much longer term effects through Krebs protein changes in gene expression. Now, what examples involve the cyclic AMP system? Well, the cyclic AMP system involves, for instance, catecholamines with all beta receptors, which are GS coupled, alpha-2 receptors, which are GI coupled, but also M2 receptors to acetylcholine, which are GI coupled, and certainly for your physio and biochem, glucagon receptors, which are GS coupled. The same diagram looks at the PIP2 system. And in the PIP2 system, very similar setup. 7 transmembrane domain receptor, classic of G protein linkage, a G protein, here called GQ, and an enzyme, here called phospholipase C. Only difference then, the second messenger's nature. Phospholipase C breaks down PIP2 into 2 second messengers. Diacylglycerol, called DAG, which are really the little lipid tails of the phospholipid PIP2, it stays in the membrane, and IP3, the phosphate head of that phospholipid, water-soluble, which goes inside the cell. IP3 in turn works on the sarcoplasmic reticulum of cell to release calcium. Calcium then interacts with proteins. We therefore have 3 second messengers in that system. In the GQ system, we have diacylglycerol, calcium, and IP3 as second messengers. We have diacylglycerol, calcium, and IP3 as second messengers. Is there a protein kinase? Well, those messengers can indeed interact with a protein kinase called protein kinase C. And protein kinase C, just like protein kinase A, can phosphorylate other proteins to turn them on or off or change gene expression. Classic examples here. Alpha-1 receptors, M1 and M3 receptors work this way. You know, those are receptors we often find on smooth muscle. And certainly you could relate right now the increase in calcium with interaction with calmodulin, resulting in constriction of that smooth muscle. Much more will come from these receptors. Right now, what you must know is receptors and how they're coupled. When we put names of drugs in there, a classic board question will be to give you the name of the drug and ask you which second messenger is increased or decreased inside that cell. Now, besides working on intracellular receptors, working, of course, through ion channels or working through GS, GI, or GQ, there are other signaling mechanisms. On your screen, you can see that cyclic GMP and nitric oxide signaling is also an important way of causing responses, but it's a very specific way of working. Cyclic GMP is produced through nitric oxide stimulation of an enzyme called guanyl cyclase. And guanyl cyclase, by raising cyclic GMP in smooth muscle, will result in a strong vasodilation. We'll review that mechanism of action in the cardiovascular section in drugs used in angina. One way or the other, anything that vasodilate could lower blood pressure or at least increase perfusion of tissues. Other receptors may function as enzymes or transporters. An example would be acetylcholinesterase being a famous target for inhibition by drugs used, for instance, as organophosphate insecticides or in Alzheimer's disease or myasthenia gravis, something we'll see in our ANS section. Transporters such as reuptake could be a major target. Think of antidepressants, drugs of abuse like cocaine. Other receptors may themselves be transmembrane enzymes. And one classic transmembrane enzyme that behaves as a receptor is a receptor for growth factor. Growth factor receptors have tyrosine kinase activity. Anything with tyrosine kinase activity is to be associated with growth. Often, it is not a question that will stem from your pharmacology there. It'll be a question out of your pathology, since tyrosine kinase activity, growth, and cell division are classic basis for neoplastic transformation of cells. Let me ask you just one in passing. You know CML, and you know CML, pathognomonic to it, a Philadelphia chromosome. That Philadelphia chromosome is a translocation, T922, resulting in a BCR-ABL protein. Well, guess what is the activity of the BCR-ABL if you want to see proliferation of cells? BCR-ABL has a tyrosine kinase activity. Still working with tyrosine kinase are receptors to cytokines. This is something you'll see more in your immuno. The tyrosine kinase activity of cytokine receptors is referred to as Janus kinase or JAK, and the targets of these kinases are transcription factors called STATs. Well, you would see again changes in gene expression relevant of immune responses and modulation. On the next tape, we'll look finally at clinical trials and teratogenicity to finish our general principles.